cell cycle drawing assignment, and it starts with just a description that we're not going to work with colored construction paper this semester, I guess. I don't think we have. And I do most of the classes I teach. And the idea is, again, try to create novelty, a different learning experience that is one that you would remember. I got going on some of this stuff when I started visiting high school biology classrooms. So when I started as a professor, I had never been trained to teach ever anything. I'd never taught anything. I liked to explain stuff to people, but I'd never really, never been in charge of a class even for an hour. And so I connected with local high school biology teachers, and I still go out to high schools and work with the teachers. And so this is something that you'll see is that high school teachers will do more of this model building than you see, I think, so far in college courses. And it's really good, novel, experiential, hands-on learning experience. And a lot of times when senior biology class and AP biology class will learn about the cell cycle, they'll build this circular model that's very familiar to everybody, and they'll build this and they'll say, look, this is a model like that. And the problem is, it's just the diagram from the book. And so, we're not going to build a model today. We're going to do a drawing, but you'll see that there are ways of doing drawings that are different. For example, when I go to high school biology classes, what we do to help understand the cell cycle is, and maybe because students are less familiar with the idea of a traditional analog clock. They see digital clocks now, and they aren't so used to this idea of, it's a repeated cycle going round and round like that. And so what we do is we start out building a linear model. And you'll see that there's pieces of this that are of value to really appreciate the cell cycle. This is what would be seen under a microscope. And so we get that on there. And what you see under a microscope is the cell cycle is just interphase, and you get the hints that maybe there's multiple steps underlying it. And during interphase, we have really important cell functions. This is really when the cell does its living. There's cell growth, there's DNA synthesis, there are all the functions, like all the things we talk about, like the signaling pathways. And there, of course, is the start of the cell cycle. So interphase is really significant, but under a microscope, it just looks like the same thing. And then under microscope, we see mitosis, and I made it the same color, lump them together, mitosis and cytokinesis. So mitosis is the nucleus divides, so that process when CDK1 and cyclin B, also called MPF, maturation promoting factor, that's when the nucleus divides, and then we have cytokinesis, that's when the cell divides. Under the microscope, you can see this, and we will talk about this in this assignment too. So you can see that mitosis is really composed of prophase, metaphase, anaphase, telophase, and we see those cell functions then during that, right? And these are part of mitosis, right? So it's nice to build physical models like this to start getting them physical and memorable. So when you recall, I remember how we pulled the mitosis apart, and interphase was just a kind of a blur. So we do that, and then we say nowadays, we see that the cell cycle is really composed of these separate steps or phases like that, right? So we just tape this on, and then one thing we have is this feature, and this is a nice feature of a physical model, and that is, you can see, look, this is what we're representing here. So we have this cell cycle, a wheel-like, circular clock-like, old-time clock-like diagram, and we can show it and physically work with it by making it linear. One thing that happens in the classes is we get going on this, and the students can chain their models together. So the class can get together at the end, and they can show multiple rounds of the cell cycle, and we can talk about how that differs in a stem cell, or what happens when a cell differentiates, like that. And so we're not going to do that today. What we're going to do is we're going to draw, but we're going to try to draw that out in a novel learning way, and so it's going to go like this. And so what you're going to do is it's really, I think, really two drawings, and you see how I taped paper together like that. And the two drawings are just different parts of showing the cell cycle like that. And so we'll start out up here and we'll say, okay, we're going to draw the cell cycle in our advanced class where it actually starts here. And then we're going to have the cell cycle progress like this across the page like that. And it's going to be composed of, we're going to have S phase here, and then G2, and then M phase, and then we go back, and this is where we'd have G1. So the start occurs in G1, but it's at the end of G1. And this is where we'd also want to represent that here at the end of G1 is where some cells spend a chunk of time, could be minutes, could be days, could be years in, G0. Okay, so that's the first part of it, just mapping the idea out, in our case, that when making a linear model of the cell cycle, we'd like to have it start at start, like that, and go on. And then what we're going to talk about in this first drawing is really to expand on this M phase part of it. So we're going to add this and expand it out, and this part of the drawing is initially some review. And so we're going to divide it up into prophase, metaphase, anaphase, telophase, and cytokinesis, like that. So prophase, metaphase, and one reason I'm doing this is I'm showing you, like, make it big. And I've said before, I think when students start doing this, they make it small partly because they're not too confident in making it big and showing off their knowledge, but also because they're used to working within one piece of paper, like that, cytokinesis. And again, you know, it is true that telophase and cytokinesis are distinct phases of the cell cycle. People put them together. They put cytokinesis in as just its M phase. But mitosis, M phase, really is this first part. And the reason that's significant is because that's when CDK1 and cyclin B are active. And that's what that means. Okay, so we want to get that in there. And then we learned that we should learn a step, something that happens in each of these steps. So chromosomes can dance is a good one. And in a lot of organisms, including humans, the nucleus breaks down. And so we've learned about that, and we know that's because the CDK1 and cyclin B is active. Then in metaphase, the chromosomes line up, and they line up in the middle of the cell, and it looks like a fence or a line, and people call that the metaphase plate, because a plate is a flat-looking object, so it's the metaphase plate. And then in anaphase, again, we can see those chromosomes, and we know there's something significant under a microscope. For the last 50 years, the chromosomes separate, okay? And then telophase, so what happens in telophase is the nucleus reforms. And we've separated the chromosome, so really this is plural. The nuclei reform, because there are now two nuclei, like that. And then also the chromosomes decondense. What does that sound like? It sounds like during telophase, the CDK cyclin B get inactive. That's when they finish M phase and they're inactivated. And at the same time, really, during telophase, cytokinesis begins. And cytokinesis is cell division. And so we learned about that before, but it's good to spread it out, learn it this way. And what we're going to do now is we're going to start putting in a new thing that we didn't talk about, and that is maybe down here we will put a drawing of a little table. And so these are major cytoskeleton components, and I'll run out of space there, but components, and we're just make a little table, and there are really 3, and we're going to give their name, and then what are they composed of? They're composed of protein monomers that get polymerized, and then what's their common role in the cell? So let's put some colors into this, and the first one we're going to talk about is cytoskeleton components that are called intermediate filaments. And they're composed up of monomers of various proteins. And we're going to put down here like a little footnote, and that is the really one category are there's some proteins called lamins, and we are learning about, for our example, nuclear lamins, and those are proteins that make the mesh that lines the inner part of the nucleus. So those are nuclear lamins that are joined together during interphase, and this is why the nucleus breaks down during M phase. And so we could put up here a little thing like this, and then all along in here, these nuclear lamins are scattered, and then they reform, and they form 2 nuclei around the separated sister chromatids, like that. The nuclear lamins form this structure, which is called the nuclear lamina. And so for our cell cycle model, there's a function for intermediate filaments. So one of the major cytochemical components are intermediate filaments, and this is the role they play. What's the component's role? And they make pretty stable, so I put this in quotes, structures like the nucleus and the nuclear lamina. And so they're kind of boring because they make pretty stable things, and the next ones are going to be very dynamic stories. And so that's our first one. And again, this is a table that you likely learned in intro bio. And it's nice to just memorize this by having a time frame where these guys play a role during the cell cycle. So the next one is going to be microtubules. And microtubules, woo, that's some spelling. Microtubules are made up of monomers of tubulin that get polymerized, and they the way to describe their function, and these functions are somewhat overlapping, but this is a good like intro bio categorization of them, is these tend to move things inside the cell. And do we have a story about microtubules in the cell cycle? Yeah, oh yeah, you bet. Microtubules make this structure, which is called the mitotic spindle. And it forms during metaphase, and it is the component of the cytoskeleton that pulls the chromosomes apart during anaphase. And when we move things, instead of being stable, these things are dynamic. And so we get a lot of things being moved around in cells as a result of microtubules being dynamic in forming and organizing and then moving things around like chromosomes. So that's a great example of that. And then we'll use this, if it's a good color, we'll use this pretty purple one. And the last one is microfilaments. And so in every Intobio textbook in the first semester, these three are listed. Microfilaments are composed of polymers of actin protein. And because of that, some scientists, like me, call these, I always try to call these actin microfilaments. And it's because these words, microtubules and microfilaments, look too similar for students when they're learning. And these play really different roles, and they're really different stories. How can we tell them apart? And one way is just to call these, for the rest of your life, actin microfilaments. And these are microtubules. Actin microfilaments remind you that these are composed of actin, and you might know of actin, a different member of the actin gene family. That protein plays a role in muscles, where it is involved in muscle contraction by the motor muscle, the ATP-driven motor myosin. And that actually is what happens here in the cytoskeleton. It's different members of the gene family of actin and myosin, but they contract, and what they do is they are really typically play a role in changing the cell shape. Like that. And so do we have an example of that in the cell cycle? Oh yeah, definitely. So during cytokinesis, we have the, let me just use a different color to get that on there. We have the cells that are going to divide, and they pinch away from each other by a process that involves actin polymers forming a string-like on the inside of the cell membrane, but a string-like structure called the contractile ring. And the contractile ring is a great name because it's a ring and it contracts. How does that contract? And the answer is, oh, there's myosin in those, and they causes the actin fibers to slide past each other. And so the actin microfilaments contract, and we get the contractile ring, making a smaller and smaller ring circle till the two cells pitch apart and cytokinesis is complete. And so, you know, a great table, but tables are, you know, something that a person really just has to memorize. It's a great way to organize stuff, but also to organize it in and with you know, in a complementary way with a more visual thing, right? So you go, yeah, oh, I see a role for nuclear lamins and the intermediate filaments here, and then a role for microtubules here, and then a role for actin microfilaments here, and they're really satisfying cell biology visual roles of that important process. So that's our first drawing, and you can see it's mostly review, but it's to lay it out. Now, remember, we didn't make a construction paper model. That would take a while. You're welcome to do that and actually create this using this kind of strategy, where you make this by making a fold-out, blow-up mitosis, M phase and cytokinesis. But you're not required to, because it would take a long time. And but it's great to spread it out to that size scale, not crunch it into a single piece of paper, but spread it out. So when you remember it, you're actually moving around like that. And you know, it's just a great timeline image. This is going to be another timeline image. And let me just show you that in the book, and I have some of the slides that our book uses. In our book, the book shows this figure, and it's a comparison of meiosis and mitosis. And we want to learn about that, but that's a visual that, you know, the artist spent a lot of time making, but I think there's a better visual to learn the comparison of meiosis and mitosis. And what we're going to do is we're going to draw them out like this. So this is like the second drawing, and again, do this drawing, take a picture, email it to me along with this drawing, and we're comparing now. Oh, this might not be a very good pen to use, so let's just go over to this one and say we want to compare. People call this mitosis, and mitosis is really just M phase. So what we want to compare is what's called the mitotic cell cycle. And that's what we've actually been learning about. People really typically just call it mitosis. And that's a misnomer because that's mitosis there, M phase. And the mitotic cell cycle goes like this, right? Well, we would draw it as start. And again, we could make this as a big colorful construction paper drawing, but it'll work out here fine to say this is start. And we are going to go to a series of steps like that, right? And instead of blocking them all out like that, for this, we are just going to draw it out simply, because we're going to go to S, then G2, then M. Then we're going to go to G1, and S, and G2, and M. Then we're going to go to G1, and S, and G2, and M, right? And on and on and again. Again, the cell cycle not as a circle like the clock running around, for the day, but a linear path to provide that visual. Because what we're going to do is we're going to compare it to, and I won't write down the details of the mitotic cell cycle, because what we really want to do is compare it to the meiotic cell cycle, and that's the cell cycle that is for meiosis, and that's called the meiotic cell cycle, and that's occurring in our gonads, and the gonads of all vertebrates, and then the reproductive cells of invertebrates, in our case the ovary and testes, and the meiotic cell cycle leads to the production of sperm and egg. So mitotic cell cycle occurs in all of our cells except the cells that produce the sperm and egg, and they produce that. So here's the meiotic cell cycle, and it goes like this. So we start at start, so we're going to have it regulated, and then we go into S phase, G2, then M phase we'll talk about, then G1, and S, and G2, and M, and G1, and we'll stop there because you'll see that the meiotic cell cycle stops when the sperm and egg form. Okay, so let's talk about how this, now again, we aren't reviewing this one, but remember what happens here. Now what happens to this one? Yep, we go through S, and so one thing we want to start doing is keeping track of the chromosome numbers. So remember back here, we would have this, because this is a G1, G0 stage, the cells are diploid, and maybe I'll just try to make that with a little different color, because that's a general terminology. And the cells are also, there's something I don't think we've mentioned, and they're referred to as 2N. Then in G2 we learned the cells have the chromosomes have replicated in S phase, and they go up to tetraploid 4N. And so here we have homologous chromosomes, one from mom, one from dad. Homologous chromosomes have the same genes, but they might have different versions, different alleles of those genes. So we inherited one version of the insulin gene from mom and one version of the insulin gene from dad on these homologous chromosomes. So the G2, of course, still has homologous chromosomes, but each of those chromosomes got replicated, got photocopied. We have exact copies, and the exact copies are called sister chromatids. And so this is what's happening. So S phase, meiosis, the mitotic cell cycle looks like the mitotic cell cycle for up till here, because this is not mitosis. This is called, and now I gotta get back to the right coloring. So this is actually called meiosis 1. So that's our first difference, because in the history of the sperm and egg producing stem cells, they were just going through the mitotic cell cycle, and it looks like they're going through the mitotic cell cycle up until here. And there are two main differences between the meiotic cell cycle and the mitotic cell cycle, and we want to pay attention to them. And one of them is just a kind of a bookkeeping story, but it's a little bit surprising, and it's quite different than mitosis. The sister chromatids stay together. So in mitosis, significantly, the sister chromatids separate, and we get two cells out of it. The second thing is that something that's called crossing over, or it's also called recombination, occurs in meiosis 1. And we just need to know this because that's different than mitosis. And if you took genetics, you would have learned a lot about mitosis, crossing over recombination. If you took intro bio, you probably learned about a little. But that's the distinction there. Now, then in this G1 now we have a different situation, because the chromosomes have gone down from tetraploid, and we've had meiosis and cytokinesis. The cell divides into two cells. So now we have two cells. We went from 1 to 2 cells, but these have half as many chromosomes and have as much DNA as the tetraploid cell, but they can't be called diploid, because we don't have these homologous chromosomes, one from mom, one from dad. We have the sister chromatids paired. So this is different, and we can't really call this diploid. Then here comes the next really big distinction between the mitotic cell cycle and meiotic cell cycle, and that is between or after meiosis 1, there is no S phase. So the cells simply go from G1 to G2, and then they go on into meiosis 2. And there's only one thing we need to learn about meiosis 2, and that is the sister chromatids separate. And as a result of that, this next G1 is the cells are haploid, and they go into G0, and they form sperm and egg. And we've gone from 1 cell to 4 cells. One diploid cell became tetraploid, and then through meiosis 1 and meiosis 2, we did separate the chromosomes. So now we're down to haploid cells, and that's called 1N. So 2N, 4N, 1N. And just up here, just to show it, we go 2N, 4N, 2N, diploid. Don't have that after meiosis 1. Then tetraploid, then diploid, and we just go 2N, 4N, 2N, 4N. And that's the distinction. That's our second drawing. Take a picture of that, send it to me, and we're all set. All right, thanks a lot.